Next scientist was Guha and Maheshwari in the year 1964. Guha and Maheshwari developed in vitro production of haploid embryos from anther of datura. In vitro means a laboratory production of haploid embryos with the help of the anther of the datura plants. It is also one of the milestone in a plant tissue culture. Next scientist was Wasil and Hilbert in the year 1965. Both of them achieved differentiation of tobacco plants. What is the meaning of differentiation? A simple cell that can be able to differentiate it into a complex one, that is called it as differentiation of tobacco plants from a single isolated cell in a micropropagation technique. What is micropropagation? It is one of the process of micro level of cell or a tissue that can be able to propagate in a laboratory condition that can have the capacity to produce several thousands of plants in a particular time. That is called it as micropropagation. This was proposed by Wasil and Hildebrand in the year 1965. Next scientist was Takeby et al. In the year 1971. Takeby et al. Regenerate tobacco plants from isolated mesophyll protoplast. Here, Takeby et al. Use isolation of protoplast. That isolation of protoplast, we have to form a regenerated tobacco plant. Next scientist was Carlson and co-worker obtained protoplast fusion between Nicotiana glucona and Nicotiana glucana and Nicotiana Longadorfi and developed first interspecific somatic hybrid. What is the meaning of interspecific somatic hybrid? Inter means different, specific means species. Here Nicotiana glucona is one of the species and Nicotiana Longadorfi is another species that can be able to, both of them that can be able to fuse with one another with the help of protoplasmic fusion. Both of the protoplasmic fusion obtain a somatic hybrid in the year 1971. It is one of the milestone. Next scientist was Melcher and co-workers in the year 1978. Melcher and co-workers developed intergenic hybrid. Inter means different, genic means genera. Hybrid between potato and tomato. Potato as well as tomato, both of them comes under the same family, Solanaceae, but the binomial was potato was Solanum tuberosum and tomato was Lycopersicon esculentum. Both of them comes under different genera, so it produces a tomato plant. It is also one of the protoplasmic fusion. With the help of that, Melcher and co-worker produced tomato plant. Next scientist was Chilton in the year 1983. Chilton produced transformed tobacco plants from single cell transformation and gene insertion. That single cell transformation and gene insertion that can be able to produced with the help of the tobacco plant. Gene insertion is nothing but insertion of genes with the help of the plant tissue culture technique, and cell transformation is one of the process of plant tissue culture technique that was produced with the help of tobacco plant. Next scientist was Horse et al. In the year 1984. Developed transgenic tobacco plant by Acrobacterium mediated gene transfer. Acrobacterium is one of the bacteria, soil loving bacteria. Acrobacterium tumefaciens, that is mainly used for the genetic transformation, especially in a commercial plant like potato. These are some of the milestones that can be able to present, or it can be in our branch of the plant tissue culture. These are all the one comes under the milestone of the plant tissue culture technique.